Welcome, ladies and gentlemen, back to this afternoon session of Science for Life Symposium. VinFuture Prize 2022, Reshaping and Reviving, wishes to find scientific solutions to help solve urgent global problems. We also hope to inspire and spread good values that science and technology can bring to all, including healthcare. Now that you've had your lunch, I'm sure you're feeling pretty healthy and ready for this session. So through the theme of this biomedical discussion, Precision Cancer Therapy, VinFuture Prize 2022 hopes to create a forum for the world's leading scientists to discuss big problems of global health, as well as its potential for development in medicine for human service. We'd like to extend a warm welcome to our distinguished guests, Associate Professor Dr. Lương Ngọc Khuê, Vice President of Vietnam's National Medical Council, Director of Department of Medical Service Administration, Ministry of Health. Welcome, sir. We'd also like to invite Dr. Nguyễn Ngô Quang, Deputy Director of Administration of Science and Technology Training, Ministry of Health. Welcome. We would also like to invite Professor Molly Schoiket, VinFuture Prize Pre-Screening Committee Member, Chair of this session. Welcome, Professor. As well as distinguished speakers and panelists who will bring us the meaningful discussion today. Ladies and gentlemen, welcome back to this afternoon session. To start this afternoon's event, we'd like to invite Vice President of the Vietnam's National Medical Council, Associate Professor Dr. Lương Ngọc Khuê, to the stage for his speech. Xin trân trọng kính mời Phó Giáo sư Tiến sĩ Lương Ngọc Khuê lên sân khấu có đôi lời phát biểu. Xin mời ông.
Kính thưa các quý vị khách quý, các nhà khoa học quốc tế trong nước, kính thưa các quý vị đại biểu, kính thưa toàn thể hội thảo. Trước hết cho phép tôi được chuyển lời của bà Đào Hồng Lan, Bộ trưởng Bộ Y tế nước Cộng hòa xã hội chủ nghĩa Việt Nam và lãnh đạo Bộ Y tế gửi tới các thầy, các cô, các nhà khoa học quốc tế và trong nước, các quý vị lãnh đạo, các anh, các chị, các bạn đồng nghiệp, lời chào trân trọng và chúc hội thảo chúng ta thành công tốt đẹp. Hôm nay, chúng ta có mặt tại đây trong khuôn khổ giải thưởng VinFuture, một trong những giải thưởng về khoa học có giá trị lớn, nếu không muốn nói là mang tầm cỡ hàng đầu trên thế giới, để gặp gỡ và thảo luận về những vấn đề chuyên môn cùng các nhà khoa học quốc tế trong nước về một liệu pháp tiên tiến trong điều trị ung thư, liệu pháp cá thể hóa. Kính thưa các quý vị, các thầy, các cô, các nhà khoa học, ung thư vẫn luôn là mối quan tâm hàng đầu của các quốc gia trên thế giới. Chúng ta đang chiến đấu với đại dịch Covid, mặc dù đã ngăn chặn và đẩy lùi. Tổng Giám đốc Tổ chức Y tế Thế giới cũng đã nhấn mạnh, trước, trong và sau đại dịch, chúng ta vẫn luôn phải chiến đấu với các căn bệnh truyền nhiễm, nguy hiểm, cũng như các bệnh không lây nhiễm, trong đó ung thư là một trong những căn bệnh gây nên gánh nặng hàng đầu tử vong trên thế giới và ở Việt Nam. Ở nước ta, ung thư cũng được coi như là một nhóm bệnh ưu tiên thuộc chiến lược quốc gia phòng chống bệnh không lây nhiễm. Những con số về ung thư trên thế giới và Việt Nam luôn cho thấy nó gia tăng ở mức độ toàn cầu hóa, đô thị hóa và những vấn đề về biến đổi môi trường biến đổi khí hậu, thay đổi lối sống lifestyle và những vấn đề liên quan trong cuộc sống hiện đại. Theo thống kê của Globalcan, năm 2020 tại Việt Nam có hơn 182.000 ca mắc mới và hơn 122.000 ca tử vong ung thư. Bệnh ung thư nói chung cho đến nay, chúng ta vẫn chưa có thuốc điều trị đặc hiệu. Chưa kể đến, đại dịch Covid vừa qua đã gây ra rất nhiều thách thức trong việc chăm sóc và điều trị bệnh nhân ung thư. Với mục tiêu giảm bớt gánh nặng gây ra từ căn bệnh này, trong thời gian qua, nhà nước Việt Nam đã luôn chú trọng phát triển các đề án, sáng kiến và quan tâm đầu tư cho lĩnh vực phòng chống ung thư thông qua các hành động cụ thể như các chiến lược quốc gia phòng chống bệnh không lây nhiễm, trong đó có giai đoạn 2015, giai đoạn 2025, chương trình nghiên cứu ung thư trong toàn quốc và các đề án chuyên biệt được các bệnh viện ung thư và các bệnh viện trong toàn quốc triển khai tổ chức thực hiện. Kính thưa với các quý vị và đại biểu chúng ta hôm nay với một sứ mệnh hết sức quan trọng trong khuôn khổ của giải thưởng VinFuture, chúng ta cùng nhau lan truyền cảm hứng mạnh mẽ đến cộng đồng và cùng nhau chia sẻ những kinh nghiệm, những phát minh, những sáng kiến, trong đó có việc tọa đàm liệu pháp cá thể ung thư hóa trong điều trị. Trong chương trình, chúng tôi được biết rất nhiều các nhà khoa học trên thế giới và Việt Nam sẽ chia sẻ những kinh nghiệm và từ đó chúng ta sẽ là những bài học để giúp cho người bệnh, giúp cho thầy thuốc và giúp cho Việt Nam chúng ta tiếp tục hội nhập trên con đường khoa học cũng như giúp cho người bệnh Việt Nam chúng ta. Từ thành công lan tỏa mạnh mẽ của mùa 1, chúng tôi tin tưởng rằng giải thưởng Vingroup năm nay cũng sẽ tiếp tục là sợi dây kết nối tri thức của các nhà khoa học trên thế giới và Việt Nam. Là nơi tôn vinh và thúc đẩy ứng dụng khoa học công nghệ, tiếp tục tạo ra những kỳ tích mới, góp phần làm cho cuộc sống của chúng ta ngày càng tốt đẹp hơn. Đặc biệt với hội thảo liệu pháp cá thể hóa trong điều trị ung thư sẽ là một trong những đóa hoa giúp cho cái công trình khoa học, giúp cho kinh nghiệm của các nhà nghiên cứu quốc tế và Việt Nam sẽ được lan tỏa và giúp cho người bệnh ung thư ở Việt Nam cũng như trên thế giới. Xin cảm ơn Quỹ VinFuture đã tạo ra một diễn đàn quốc tế đầy sáng tạo, cởi mở. Cảm ơn các nhà khoa học quốc tế và trong nước cũng sẽ tìm ra những vấn đề giúp cho người bệnh ung thư nói riêng và cho sức khỏe người dân trên toàn cầu nói chung. Chúc cho tuần lễ giải thưởng Vingroup, VinFuture và tọa đàm của chúng ta hôm nay thu được kết quả tốt đẹp nhất. Kính chúc sức khỏe tất cả các quý vị đại biểu và các bạn. Xin trân trọng cảm ơn.
Xin cảm ơn Phó Giáo sư Tiến sĩ Lương Ngọc Khuê. Thank you very much, sir. Ladies and gentlemen, we'd now like all of you to focus your attention on the screen for a short video.
On November 15th, 2022, the world's population officially reached 8 billion people with challenges in food, health, and development. In the field of medicine and healthcare, cancer is one of the global concerns with alarming numbers which have been causing great losses to humanity. Almost 10 million people die from cancer annually, with nearly 1,700 deaths per day. According to annual cancer reports, global spending on oncology treatment reached 187 billion USD in 2021. It is also worth mentioning that the phenomenon of a resurgence in cancer is taking place in some parts of the world, with 1.19 million new cancer patients and 396,000 deaths between the ages of 15 to 39 in 2019, and keeps raising at 0.38% annually all over the world. This is considered a huge challenge for modern global medicine. To solve this challenge, besides advanced cancer treatment methods, scientists and doctors have been developing a new therapy, immunotherapy. And in 2018, the world witnessed a revolution in cancer treatment, with the Nobel Prize in Physiology or Medicine honored 2 awards for research in this field.
I'll never forget the first day we treated when the first patient was infused. It was the culmination of many years to put all the pieces in place. But I imagine it wasn't much different than an astronaut the first time the space shuttle lifted off. You don't know if it's going to blow up or if it's going to come back intact. It was a wild ride, that first patient, who had many weeks of fever and became very ill and nearly succumbed. But he made it and he's fine now. So it was a moment that's forever etched in our minds.
Up until now, immunotherapy has made remarkable progress, becoming the 4th pillar in cancer treatment, with promising potential that attracts scientists all over the world, with the hope of bringing a better life to cancer patients and their families so that cancer is no longer a threat to people.
People used to say that we were living in the antibiotic age and then that we were living in the vaccine age. Are we now living in the cell therapy age?
We're just beginning to live in the cell therapy age. As we enter and manipulate more and more cells in the cartilage, cells in the pancreas to cure type 1 diabetes, potentially cells in the brain to cure depression and schizophrenia. We are living in an age where cells have become an amenable unit of therapy.
Under the theme Reviving and Reshaping of the VinFuture Prize 2022, aiming to find outstanding science and technology achievements, today we will discuss precision cancer therapy in the 3rd session of the Science for Life Symposium with the world's leading scientists.
You've just seen data of cancer in the previous video. Cancer is now one of the most common global diseases, showing signs of rapid increase, causing significant losses to society and threatening global health. You've also seen an overview of immunotherapy, which is considered to be revolutionary in cancer treatment, with promising achievements on cancer cases so far. That's why we're here today to talk about this inspiring topic. The biomedicine session includes 3 main parts: presentation, panel discussion, and Q&A, focusing on advanced applications of cell therapy and gene therapy in cancer treatment, successful case studies, as well as the opportunities for Vietnam. To begin our event this afternoon, let's welcome Professor Jae-jung Lee, CEO of Vaxcell Biotherapies, to give us some latest updates on clinical application of innovative cellular immunotherapy in cancer treatment. He is also the Professor of Department of Hematology at Cheonnam National University Hwasun Hospital and Cheonnam National University Medical School, Director of Research Center for Cancer Immunotherapy at Cheonnam National University Hwasun Hospital, and the Chair of Cell Therapy Immunotherapy Board, the Korean Society of Blood and Marrow Transplantation, Republic of Korea. Professor Lee, the stage is yours.
Thanks, MC, for kind introduction. Good afternoon, everyone. I am very pleased to present at this symposium organized to celebrate the VinFuture Prize 2022. So, Vaxcell Bio was started by university research in 2010 and aims for the best in class in anti-cancer immunotherapy. We have 3 platforms such as NK cell therapy. The second is CAR-based immunotherapy. The third is anti-cancer immunotherapy in pet dog. The killing mechanism of NK cells are ADCC effect, direct tumor killing, and immune cell activation. Previously, we conducted a phase 1 clinical study using local regional high-dose autologous NK cell therapy with hepatic arterial infusion chemotherapy in patients with locally advanced hepatocellular carcinoma patients. This study was published in Frontiers Immunology in this year. The objective response rate was 63%, and disease control rate was 81.8%. This patient had huge hepatocellular carcinoma and achieved complete response after our treatment and maintained CR over 2 years. Actually, 5-FU is an antimetabolite. Cisplatin is a chelating agent. This chemotherapy agent have interesting another immunomodulatory function to upregulation of NKG2D ligand for inducing synergistic effect and combined with NK cell therapy. Based on our phase 1 clinical trial, we entered phase 2A clinical trial to patient with the advanced hepatocellular carcinoma patient. So we treated patient with 2 cycles of NK cell therapy, or total of 10 NK cell infusion. In our phase 2A preliminary data, the overall response rate was 66.7%, including 33% of CR, and disease control rate showed 100%. This case showed have multinodular hepatocarcinoma in liver and achieved complete response after our treatment and maintained CR over 2 years. Our treatment of NK cell don't have any significant adverse event. Although this phase 2 study is still in progress, the preliminary data suggests that the combination of hepatic arterial infusion chemotherapy and local regional infusion of NK cell therapy is a safe and effective treatment modality for locally advanced hepatocellular carcinoma patient refractory to the standard treatment. We performed emergency clinical trial on patients with liposarcoma with lung metastasis and hepatocellular carcinoma with lung metastasis. The NK cells were combined with checkpoint blockade such as pembrolizumab and nivolumab, and the patient achieved complete response. As a follow-up study, we tried to the possibility of NK cell therapy in advanced pancreatic cancer. The combination treatment of NK cell and modified FOLFIRINOX showed strong cytotoxicity in vitro and potent anti-tumor effect in vivo pancreatic cancer mouse model. Currently, we are preparing a clinical trial by combination of NK cell and modified FOLFIRINOX in advanced pancreatic cancer patient. In small cell lung cancer, PD-L1 expression is very low. When lung cancer cell lines were exposed by NK cell, the expression of PD-L1 is significantly increased around 90%. Thus, the NK cells were first treated, and then NK cell and atezolizumab PD-L1 antibody were treated. There was strong anti-tumor effect against small cell lung cancer mouse model. Based on this study experiment, we are planning a clinical trial for the combination of NK cell and atezolizumab in patients with extensive stage lung cancer. Activated marrow-infiltrating lymphocyte meals are produced by expansion and activation using bone marrow mononuclear cell from myeloma patient. This meal contained the increased population of CD8 positive T cell, especially in niche of central memory T cell. This activated meal showed strong cytotoxic effect against autologous CD138 positive primary myeloma cell from the same patient. We try to develop CAR meal by transducing CAR into meal. CAR meal also enriched central memory T cell like previous meals. This CAR meal show more strong cytotoxic effect than meals against CD138 positive autologous primary myeloma cells. We are developing the next generation monobody-based anti-EphA2 CAR T cells targeting solid tumors such as pancreatic cancer, ovarian cancer, prostate cancer, colorectal cancer, and gastric cancer. This slide shows our CAR T cells have potent killing effect on these solid tumor in vitro and in vivo experiment. In summary and conclusion, the primary data suggests that the combination of hepatic arterial infusion chemotherapy and locoregional infusion of NK cell therapy is a safe and effective treatment modality for locally advanced hepatocellular carcinoma patients refractory to the standard treatment. It was suggested that NK cell therapy can induce synergistic effect through combination therapy with several anti-cancer agents. Therefore, the importance of combination approach is being emphasized more for successful NK cell therapy. Meals can easily be expanded from bone marrow mononuclear cell to demonstrate enhanced antigen specificity and enriched memory T cells for targeting autologous primary myeloma cell. In our preliminary experiment, CAR meal for targeting multiple myeloma and the monobody-based anti-EphA2 CAR T cell for targeting solid tumor are expected to become powerful treatment tool in the future. I'd like to thank all my members at Research Center for Cancer Immunotherapy of Jeonnam National University Hasun Hospital and all members for Paxel Biotherapies. Thank you for your attention.
Thank you. Thank you, Professor Chejong Lee. Thank you very much, sir. Next, we would love to give the floor to Professor Bruce Levine from the University of Pennsylvania, who will walk us through autologous CAR T-cell immunotherapies. Professor Bruce Levine is the Barbara and Edward Netzer Professor in Cancer Gene Therapy. He is the founding director of the Clinical Cell and Vaccine Production Facility in the Department of Pathology and Laboratory Medicine and the Abramson Cancer Center, Perelman School of Medicine, the University of Pennsylvania. He's also co-inventor of the first FDA-approved gene therapy, Kymriah, chimeric antigen receptor T-cells for leukemia and lymphoma, licensed to Novartis. Please welcome Professor Bruce Levine. The stage is yours, sir.
Thank you very much. It's a pleasure to be here, and I must thank the organizers for the invitation to share some of our work with you and express thanks to the VinFuture organization. So first, I have a conflict of interest due to intellectual property and patents in the field of cell and gene therapy, And that conflict is managed in accordance with University of Pennsylvania policy and oversight. So let's think about cancer immunotherapy. The immune system evolved to tell self from non-self. Cancer is derived from our own cells that don't stop dividing or don't die. So how can the immune system recognize cancer if it is yourself? Well, in reality, cancer-specific immune cells are extraordinarily rare. It's like looking for a needle in a haystack. It's actually like looking for a dull needle in a haystack, because cancer has developed ways to suppress from and hide from the immune system. So the natural T cell receptor is shown on the far left. It's a heterodimer that recognizes antigen in the context of a self molecule, MHC. We can isolate those rare cancer-specific T cell receptors, but they must be matched to each individual's tissue type. An alternative is to create a chimeric antigen receptor, or CAR, that is derived from an antibody on the outside of the cell and T cell signaling domains on the inside of the cell. So this is off-the-shelf way of redirecting T cells. So I showed you a cartoon. What happens under the microscope? We have colored the tumor cells green. In this case, the CAR T cell is in the bottom left. And what you're going to see over the course of 8 seconds is recognition, activation, the secretion of cytotoxic granules that puncture holes in the cell membrane, and that tumor cell is going to explode. So here we have the recognition, and now you see that cancer cell exploding. That CAR T cell is a living drug. It will go kill the next tumor cell. It will divide, and its daughter cells will also kill tumor cells. This is a force multiplier. How do we make these CAR T cells? We do it outside of the body. We collect immune cells from the patient. At the manufacturing facility, we can isolate the cells of interest. We deliver the gene using a virus as a vector. And that virus is actually disabled HIV. It cannot cause disease. It only delivers RNA that is reverse transcribed into DNA that permanently integrates into the genome of the cell. So that cell and every daughter cell has that gene. We grow the cells up in a bioreactor for several days, 9 or 10 days, and then we harvest and formulate and cryopreserve. Well, that's a very simple version. In reality, we have to think about how we collect the cells, how we wash them, how we transport them, how we manufacture the viral vector, how we do the formulation and filling, and all of the quality control assays that must be reviewed prior to infusion. Now, I want to take you to April 2012. Actually, these may be out of order slightly. That should have said August 2010. My apologies. We started in chronic lymphoid leukemia in patients who had relapsed and were refractory to all available treatments. This is our first patient treated in August 2010, Bill, and up top center you can see his bone marrow. Everything that is brown is leukemia. 6 months after treatment with his own engineered CAR T cells, his bone marrow is clear. There's no leukemia. Between 1.3 and 3.5 kilograms of leukemia in the first 3 patients were obliterated in the several weeks after infusion. Now, Bill has chronic lymphoid leukemia in this picture and no longer. Next, in April 2012, we wanted to move to acute lymphoid leukemia, a very rapidly dividing leukemia, to see if the CAR T cells could outrace the disease. This is the first patient, Emily. She was diagnosed in May 2010. She relapsed twice, came to Children's Hospital of Philadelphia and received a high-dose chemotherapy regimen that would kill an adult, and only 1/3 of kids make it through. She did make it through, but in this picture, her bone marrow has 60% leukemic blasts. She has lesions in her kidney, liver, and spleen, and she is not far from her demise. And here's what happened up top. We're looking for the CAR T cells on the bottom. All the dots are leukemia. 23 days later, we see a massive expansion of the CAR T cells in the bone marrow, and the leukemia is clear. In 23 days, minimal residual disease. Well, with these activated CAR T cells, they secrete chemical messengers of inflammation that leads to a cytokine release syndrome that can be life-threatening. That is significant, but we have learned how to manage it. It's a short-term toxicity. I'd like to remind you that the conventional treatments for childhood cancers carry significant long-term effects in multiple organ systems. Today, we have 6 approved CAR T-cell therapies from 3 manufacturers. They are approved for indications in leukemia, lymphoma, and myeloma. We also have gene-modified stem cell therapies in adrenoleukodystrophy and in thalassemia, and there will be more approvals in the next year. This type of medicine is coming. I want to address how we think about potency and teaching every T-cell all that it can be. Because we need to work on potency improvements. We have patients that don't respond. How can we change the way that we activate the T-cells, the way that we add cytokines to the culture, to work on changing the metabolism through transcriptional regulation and doing gene knockouts to enhance potency. We are in the era of synthetic biology where we can engineer CAR T cells that function in a logic-gated fashion with and or not gates. We can engineer checkpoint-resistant CARs, safety switches, and conditional CARs. And we need all of these tools in solid tumors because solid tumors have proven to be more challenging because of antigen choice, antigen escape, and trafficking. Now, CAR T cells, as they are approved, are expensive therapies. How do we think about improving that? How do we think about manufacturing faster CARs beyond the 9 or 10-day cultures and 3-week turnaround? Well, here are experiments from a collaborator of mine at the University of Pennsylvania who compared our 9-day CARs to 5-day CARs and 3-day manufactured CARs in a mouse model and showed that the 5-day CARs were more potent than the 9-day CARs and the 3-day CARs were more potent than the 5-day CARs. And this is the basis for a clinical trial that is now enrolling patients at the University of Pennsylvania. And this has a twist. It has a potency enhancer because beyond a CAR, it also has a gene that encodes the cytokine, the tumor-killing cytokine, IL-18. Just 9 days ago at the ASH meeting in New Orleans, this data was presented in 8 patients. And these are patients, 7 of the 8, that had relapsed after prior CAR T cell therapy from one of those commercial CAR Ts that I showed you. And we have very encouraging responses, a 100% overall response rate and a 57% complete response rate, again, in a population that had already failed previous CAR T cell therapy. We have a 2-day process being presented by Novartis with encouraging results and engraftment and persistence that mirrors what is seen with the current commercial CARs at a 25-fold lower dose. We also have a 1-day CAR T cell therapy from a company in China. Now, how do we think about the quality control and release testing? The conventional testing that we do is shown on the left. But for a 24-hour product, how do we think about a CAR expression where the gene may not be fully integrated? How do we think about vector copy number, replication-competent lentivirus, and especially potency for a product that is, in fact, still changing? Well, what if we go even shorter than 1 day, and we make those CAR T cells in vivo, and we use the same technology that is used in the mRNA COVID vaccine with a lipid nanoparticle encoding mRNA. Instead of coding for spike protein, it encodes for a CAR. And we direct those lipid nanoparticles using an antibody conjugated on the surface. The preclinical data for this approach is published earlier this year in Science. Well, CAR T cells are moving beyond oncology. We have clinical trials and results in HIV and autoimmunity, in fibrosis, as that Science paper showed, and potentially CAR T cells for aging. Now, in the final section, let me just briefly touch on responsibility and ethics and separating hype from hope. This is an article that appeared in Science magazine over a decade ago about the so-called stem cell clinics that are selling cures for everything from Alzheimer's to bone disease and other diseases. One cell treats all. Can that really be true? What really struck me is the quote from this patient who says, "I don't know the science behind my miracle cure and I don't care." Well, what message does that send about scientifically and clinically validated development of cell therapies? These are some advertisements from one of these clinics that appeared at a meeting I attended in January 2020. Exosomes to enhance your sex life, to grow hair, you name it. I will set up your business to sell stem cells from your storefront. As we integrate this new technology, we have to be mindful of bad actors that will co-opt these terms and try to deceive patients. I'm the immediate past president of the International Society for Cell and Gene Therapy, and we have a checklist to evaluate, is this a scientifically and clinically validated therapy? What are the things that you should look for? And I won't read through the list here, but you can see some of the things that we look for to distinguish the validation from the pretenders. Some critical path issues for wider patient access. We need to work on enhancing potency, especially in solid tumors. These therapies are complex to manufacture. How do we integrate automation and shorten the culture? We need education and training at all levels. That's what you're doing at this symposium, and that's what we do through the International Society for Cell and Gene Therapy. And finally, it may be difficult to read, but we need to recognize that these therapies are financially complex. How do we integrate predictive biomarkers, and how do we link a value-based payment that links the payment to outcomes? Well, I'll close with two heroes and two stories. You met Emily in the opening movie, and I showed you on the day of her CAR T-cell treatment. She is 10 years cancer-free, an honor student, a senior in high school. Sophia is on the left, and Sophia initially responded, but then she relapsed with an antigen-negative disease. So we need to remember both stories so that we work hard to make sure that every patient possible has a successful outcome. Emily's story is so unique. There is a full-length documentary film made about her that premiered at the Tribeca Film Festival and has been playing around the country and the US. It's not yet available internationally, but I hope you have the chance to see it soon. The Emily Whitehead Foundation had a gala event where they invited CAR T-cell kids from all over the US. This is about 24 kids who probably would not have made it had they not been treated with CAR T-cells. And finally, I'll close and recognize that I'm a representative of a very large team led by Carl June at the University of Pennsylvania. It truly does take a village to make these therapies happen. So I thank you for your attention.
Thank you very much, Professor Bruce Levine, for your information on CAR T-cells therapy, as well as its applications and case studies. Thank you. Following the two presentations given by our speakers, we now invite you to the discussion session, where more related topics will be shared and discussed by our guests. Please join me to welcome Professor Molly Schoikhet, VinFuture Prize Pre-Screening Committee member, to the stage to moderate the discussion. Professor Schoikhet is the Michael E. Charles Professor in Chemical Engineering at the University of Toronto. She is also a world leader in areas of polymer synthesis, biomaterials design and drug delivery in the nervous system, as well as 3D hydrogel culture systems to model cancer. I would also like to have the presence of distinguished guests as our panelists today. Please welcome back to the stage Professor Jae-Jung Lee and Professor Bruce Levine. Professors, welcome on stage. and gentlemen, we have three other panelists. Dr. Chi Lê Phương, Chief Medical Officer at AstraZeneca, Vietnam. Prior to this role, he held numerous positions, including Regional Medical Lead for Inflammatory and Infectious Diseases, Asia Area Medical Council Lead, and Global Leader in Quality Management and Medical Standard for Oncology. Welcome. Next up, we have Professor Ha Văn Vũ, the Percy F. Smith Professor of Mathematics and Professor of Data Science at Yale University, Scientific Director of Big Data Institute, and VinFuture Prize Council member. He is well known for his work in additive and probabilistic combinatorics, random matrix theory, and their applications in computational science. Welcome, Professor. Please also welcome to stage Professor Mark Stephen Cohen, Dean of the Carle Illinois College of Medicine at the University of Illinois Urbana-Champaign, Senior Vice President and Chief Academic Officer of Carle Health. He has been running an NIH R01-funded translational oncology laboratory for the last 15 years, working on developing novel therapeutics and diagnostics for treatment and staging of advanced cancers, as well as new programs in tissue engineering, AI, and machine learning in healthcare decision-making. Please kindly give a round of applause to our panelists. And Professor Choiquette, the stage is yours.
Thank you. Globally, we are faced with challenges in health and access to the best health solutions. In this session on health, we focus on cell therapy. The field of study first emerged with the discovery of hematopoietic stem cells. Those are blood stem cells, in 1961 by Tillan McCullough in my home country of Canada. Since then, we have understood that there are adult stem cells in many of our tissues and organs that can be stimulated to promote repair. In 2006, the Nobel Prize in Medicine was awarded to Yamanaka and others for understanding that adult cells could be pushed back developmentally to be pluripotent, thereby serving as a source of stem cells without the ethical dilemma associated with human embryonic stem cells. These discoveries, coupled with so many others, have opened opportunities for the treatment of diseases and disorders not previously thought to be even possible. It has sparked our imagination to do things differently. And that's what we will be here to discuss in this session, as we focus primarily on cancer and how we can manipulate our own cells to change our destiny. Fascinatingly, as you just heard, we can take cells out of our own bodies, transform them, and then put them back into our own bodies to kill the very cells that are trying to kill us. The use of autologous cells, that is, cells that come from us, is powerful because we should not reject our own cells. And thus, with minimal manipulation, these cells are accepted as native and can stimulate our immune cells to do their job and kill the cancer cells. This has truly transformed cancer treatment, at least in liquid cancers. Now, with new research, we are learning how to transform these cells in us, which should make the process enormously less complex and less expensive. I am excited to lead the discussion with our esteemed panel. We have 3 big topics to discuss, and then we encourage questions from the audience. So, topic number 1. There have been numerous innovations that advance and improve cancer treatment. But as we saw in the opening film, cancer incidence and deaths are still increasing. In fact, you might say that surgical removal is still the best treatment. So what can make us more optimistic at the moment? What are the recent advances in cancer diagnosis and treatment? And what are the most promising directions in cancer research? So that's a lot for one topic, but we'll break it down into a series of questions. So I start with Professor Cohen and Professor Lee. How has immune cell therapy given new hope to cancer patients? And in parallel with treatment, what are the novel advances in diagnosis and prognosis that bring new hope to cancer patients? So maybe Professor Lee, you will start, and then Professor Cohen.
The recent development of translational research, the various cancer immune cell therapy have been emerged in clinical application. The most representative one is NK cell therapy and the CAR T cell therapy. In case of NK cell therapy, it is very high advantage. It's very easy manufacture and low cost to applicable broad range of cancer. And in case of CAR T cell, is very potent, already presented by Dr. Ryan. But in case of CAR T cell, have very limitation in developing country due to high cost and some kind of complication. Anyway, this kind of immune cell therapy is in the future hope to cure in cancer. Yeah, that is one direction. In the diagnosis and the prognosis, the early diagnosis will be very important in the field of cancer immunotherapy in the future. And although the prognosis is very improved to refractory and relapsed cancer patient, however, it is still a problem to cure. So in the future, we need more new innovative treatment to cure cancer. Okay.
And I think, just that word cure, until immunotherapy, we never thought of the word cure and cancer together. And so that in itself is fascinating. Professor Cohen.
Sure. As a cancer surgeon and researcher, I think there's a lot of new hope in the therapies that are coming out, and specifically around immunotherapy and CAR T therapy. What's exciting is the thought of having autologous treatments that have lower toxicity for patients. And even now, we're hearing about new opportunities in engineering these cells to be more specific for solid tumors. An example is a recent article that came out in Science this past week looking at several different DNA sequences that can be wired into CAR T cells and allowing these genes that can be switched on and off based on exposure to different chemotherapies. To think about having the ability to control when T cells release their interleukin stimulation can be very exciting, and we think about opportunities for reducing T cell exhaustion and boosting their efficacy. I think around diagnosis in cancer, there's a lot going on with molecular diagnostics that are very exciting as well. When we think about the different modalities with circulating tumor DNA, circulating RNAs, even exciting new things like exosomes and extracellular vesicles, where we can identify tumors and progression of disease much earlier than ever before, as well as response to therapy. So I think there's a lot of hope in the future, and I think these technologies are going to continue to evolve.
Thank you. So Professor Levine, will bring you into this conversation, and feel free to add to that discussion, but I also want to get your thoughts around, what's next. So you showed us how the FDA approved CAR T cell therapy for blood cancers in 2017. And can you give us some perspective on, when will we see other CAR T cell therapies approved for other cancers, such as solid cancers? And then how CAR T cell therapy will be approved for treatment more generally. And you talked a little bit about that in your presentation, but maybe you can expand on that. And then I've got another easy question for you.
Okay, and I think we have about a minute to answer all of that, right? The answer for what's next is, I think in 2023, in the US, we're going to see approvals for at least 2 or maybe 3 new cellular therapy products. One will be a so-called tumor-infiltrating lymphocytes. Another will be a multivirus specific T-cell product, and then we'll see what's coming next. To answer first other diseases, and then I'll take on solid tumors, there is some very exciting work coming out of investigators in Germany looking at autoimmune disease, specifically lupus, using T cells. Actually, they target the same antigen on B lymphocytes as we use to target cancer. And there are studies in other autoimmune diseases as well using a flavor of this technology. Solid tumors have these challenges. Well, I should say the blood cancers maybe have an advantage, and the target is expressed only on B cells and B cell leukemias and B cell lymphomas, and that's CD19. BCMA is expressed in multiple myeloma. Each of those targets is really not expressed on other tissues. In solid cancers, we have targets that are brightly expressed on tumors, but usually they're dimly expressed on other tissues. So then you need to think about, how do you distinguish between the tumor and the other tissue? And we can do that integrating some switches, maybe some control molecules, some small molecules to turn the cells on or turn the cells off. And we have the issue of potency. Solid cancers are much more immunosuppressive in general than the leukemias and lymphomas. And then we have the issue of trafficking. You need to get inside a solid tumor. And many solid tumors, particularly pancreatic cancer, wall themselves off. They have fibrotic tissue and makes it very difficult to traffic inside the tumor. Now, we can also address that by directly administering rather than intravenous to perhaps inject near the tumor. But it may not get all the tumor. Maybe it triggers a chain reaction. Another way to address this challenge of potency is to incorporate additional therapies, perhaps with a virus that is designed to kill cancer and releases some additional antigens, perhaps in combination with radiation therapy or checkpoint antibody therapy. So we have many approaches in investigation, and we have many groups around the world, both in academia and industry, looking at this, and it's going to be an exciting time coming in the next few years.
Yeah, it does sound exciting. And one thing that we'll come back to later in the discussion is something that you just said about that collaboration between industry and academia, so and government, of course. Those three groups have to come together to really change the world. The other part of that question is really around cost. And, you know, this immunotherapy and CAR T-cell therapy is still very high in terms of costs. And so what do you think we can do, or what do you think will happen to reduce the cost?
Yeah, so first let me frame that in a number of cancer therapies, the patient needs to continue on that therapy to maintain a response, or to be on that therapy for a long time, or as I mentioned in pediatric chemotherapy, there are significant long-term toxicities. So there's cost embedded in there that might not be apparent in the near-term cost, but it carries a long-term cost. Yes, CAR T-cell therapy is expensive, but it has long-term durable benefit. But how, to your question, do we think about bringing down this cost? I mentioned in the presentation integrating automation. I mentioned the possibility, perhaps in the medium to longer term, of generating these therapies in the body by administering lipid nanoparticles or other types of nanoparticles, so that's an option as well. But we're also advising a company in India called Immuneel on how to reduce the cost of CAR T-cell therapies. And they have a first clinical trial open right now But you can recognize in India with 1.4 billion people, to provide access, you really need to bring down the cost. So we're looking forward to what we're going to learn from India on how they bring down the cost, as well as all of these other methods, including automation.
Well, that's really exciting to hear, actually. That's fantastic. Thank you. So now, Dr. Tree and Dr. Vu, I'd like to bring you into the discussion, and Professor Cohen, feel free to add if you'd like to. The question is, to what extent will precision cancer treatment evolve in the next 10 years in industrial aspects? So that's definitely for you, Dr. Tree. And then Professor Vu, and I know Dr. Tree, you'll have some thoughts, and Professor Cohen as well, on what will big data and AI contribute to this process? So first, we'll start with you.
Yeah, so as we can see, the development of precision medicine and also some achievement that we got from immunology, oncology. So in the industry point of view, in the next 10 years or even decades, what we are thinking, which will be evolving, would be firstly on the strategy that we are looking at the disease, especially the cancer. So we may have more clinical trials and even a solution for early detection of cancer, where we can see there's a potential for cure. And with the current modalities that we are having, for example, like IO and other targeted therapy, we are thinking of some combination of the current modality we are having for the treatment of cancer. In terms of platform, what we are thinking would be still, again, on tumor drivers and resistant mechanism. But then immunology oncology would be still in the pipeline. Next thing would be on antibody drug conjugate and cell therapy, of course. We see much of the achievement in this area. For technology, what I'm seeing would be a very big shifting and focus on building big data and AI. Implementing AI and machine learning in the very beginning phase of drug development, and also in the later stage of the drug development cycle. For example, like applying AI and machine learning in designing clinical trial, identifying the appropriate population that we can expedite the recruitment phase of the clinical trials. And last but not least, we are seeing of the trend in whole genome sequencing, or even a routine genetic sequencing in the clinical practice, because that can bring lots of data and can, again, support back to the secondary research and also some other generation of new concepts to support the R&D better.
Thank you. Professor Vu, would you like to add about big data and AI?
Yeah, sure. Thank you very much. Yes, so yeah, we have heard about these terms a lot recently, like big data and AI, and they sound like magic things that can do everything exactly. So what can they really do? I mean, we have data around us, and they become bigger and bigger by the days. And luckily for us, in the last like 15, 20 years, there are mathematical tools and also hardware development that make it possible to analyze them. And now the thing that we are very good at, looking at it as statistician or from the machine learning point of view, is to detect pattern in data, correlation in data, clustering data. So that is, or classification of data. Now that is the general things that a computer scientist or a statistician can do without knowing what the data is. Now, if you look to the health data, or I mean in cancer in particular, you can see well, at least two kinds of big data. One is a collective one, I mean, a files of hundreds of thousands of patients. And all the data about these patients, not only cancer, but his way or her way of life, other diseases, what he ate or how he grew up. And the second thing is that the personal data, as Dr. Chi has mentioned, like genomic sequencing data or MRI data, they are also very big inside. And with this big data and the mathematical and computer science tool we have, we can do all the things that I just said. We can find potentially find correlation between treatment and diseases. So like which group of people have the best treatment, what kind of treatment is the best for which group of people. That is a clustering problem that I talk about. Or we can differentiate between a normal cell and a cancer cell. That is a classification problem that I talk about. From my point of view, they are just 0-1 sequences, as the computer see. But you have a lot of interpretation for them. So at Yale, we have a new founded Institute for Foundation of Computer Science, and we really want to bring the people in practice, like medical school or even political school, together with the mathematicians and data scientists, so that we can, avoid the language barrier and find the common purpose that we can, to do something useful.
Yeah, I think that's a really good point. That idea of being able to speak each other's language, like even from the mathematicians and the biologists. Did you want to add anything?
I think the other two speakers pointed out some very important aspects of machine learning. That first, the algorithm has to be very well done, and the data has to support the algorithm to really train it. And getting these different types of data sources, whether it's genomics, RNA, thinking about exosomal data, epigenetic data, all of that has to be taught into the algorithm in a way that produces meaningful results. And so as we think about the future of where this is heading, I think it's super important to recognize how we can all work together to create the databases that are going to be meaningful to help answer some of these important questions, and then translating that into identifying both diagnostic opportunities as well as new biomarkers and therapeutics.
Wonderful, thank you. Well, we're now going to switch over to topic 2, which is focused more on research and development. So what are the strategies and technologies that will accelerate the development of precision cancer therapy? So Professor Levine, how can we further accelerate the process from basic research to clinical trials to new cancer therapies, such as CAR T cells? What is the biggest barrier to the process, and how can it be overcome? And then I'll get you to talk a little bit about the government strategies and policies that have boosted the development of advanced therapies, and perhaps you can give some examples in the US, but other countries as well.
Yeah, so I'll give an example of a new technology that has accelerated testing these new therapies, which is CRISPR. And just a decade ago, CRISPR was described and the application to mammalian cells, and now we have clinical trials, both ex vivo using CRISPR to knock out genes or insert genes, but also in vivo to directly deliver the machinery to change genes. So that's been a very rapid evolution. I'll pick a second issue, which is training and education. Now, I mentioned we have this symposium today. We have professional societies, but this field has been growing so rapidly that there is a severe shortage of expert and trained staff at all levels, from the technicians who manufacture and test the cells, to the laboratory directors, to the program directors, to the hospitals who have the personnel to intake these procedures to the clinicians. So we really need to work on training the trainers and expanding that so that we have people at all levels that are able to develop and manage these therapies. Now, you mentioned the government, and maybe I'll take the third part of my answer relates to the regulatory bodies and their expertise. We have some regulatory bodies that do not have regulations in place to address the receipt of applications to conduct clinical trials, to be able to review applications for approval of these therapies. Then we have to think about the people who are working at these regulatory bodies. Have they seen these technologies before? So the education cuts across many levels. We need to work on educating the regulators and people who will take these positions at the regulatory bodies. And I make the analogy to think about 120 years ago with this newfangled machine called an automobile. Where did the dealerships come from? Where did the mechanics come from? Where did the petrol stations come from? So we're really building this new pillar of medicine at many different levels.
Fantastic, thank you. So I want to keep you in this conversation, but invite Professor Cohen perhaps to start us off. What new strategies and technologies will accelerate the development of cancer immunotherapies?
So I think there's a real opportunity when we think about some novel approaches to cancer therapy that involve organoids and 3D printing. I think there's work that we're doing at our center looking at lentiviral transfection systems where we can take autologous mesenchymal or adipose-derived stem cells and transform them into other cells that can be functional organoids, and then create 3D printed scaffolds that you can implant them. I see us in the next several years being able to replace organs with 3D printing, and I think that's going to be a reality before we see the 2030. But also thinking about how these models can be used in silico to really create opportunities to really think of precision therapy much more strategically. So creating digital twins of tumors by using omics and digital sampling of tumor cell geography to really then put it through a variety of different targeted therapy sequences and using machine learning, understand what's the right precision approach to that specific tumor. So I think we're going to see a lot of things that can accelerate decision-making around cancer therapy.
Yeah, thanks. And I think this idea of models of disease, whether they're in silico or having better models of disease in vitro in the lab to study it and to come up with better therapies is critically important. Professor Levine, would you like to add to that in terms of strategies and technologies that you already talked a little bit about the CRISPR-Cas9, but just to accelerate the development of cancer immunotherapies?
Yeah, well, maybe I'll start with where we were in the mid-90s when some of these first clinical trials were starting. And we had some tools and equipment and reagents, and often they were borrowed from other technologies, or maybe they were research grade. What we've seen over the past couple of decades with the clinical results and the commercial approvals is there has been a huge investment by tools and technologies companies to develop equipment to come out with new reagents that we can then translate into the clinic. So I think that's a ripple effect of what we've seen in the research progress and then the clinical progress is we have this investment from these tools and technology companies. We also have that demand because there have been companies spun out and companies formed to develop and now sell these commercially available therapies. So that demand, if you will, has created the supply of these new tools.
Well, so that leads just perfectly into the next question, and I'd like to keep you in the conversation, but also invite Professor Lee and Dr. Tree. So if you could address, how does cross-sectoral collaboration between public research institutes, the private sector, and maybe even government, enable research and development of novel cancer therapies? So maybe, Professor Lee, given that you're in the academic sector, you've started a company, maybe you can talk a little bit about that entrepreneurial spirit that you have, and that I guess is important.
For example, I studied cancer immunotherapy in hospital for 22 years, and then we started BioVenture company 10 years before. So the meaning is in Korea, we changed the trend of research. Previously, our research focused to publish the paper to the international journal. So 10 to 15 years ago, we changed completely to focus into translational research to develop new product rather than paper. So if the researcher develops new therapeutic tool, it's needed to translate or license out to the BioVenture company to develop product. That is very important to enhance the medical the development, and I also it's the same in Vietnam. Yeah.
Thank you. Dr. Tree, would you like to add to that about that cross-sectoral collaboration?
Yeah, so in the point of view of a pharmaceutical company and also of the industry, what we are seeing from a cross-sectoral collaboration would be very useful and a key driver to help us gain success in our development program. So we bring together almost the resources that we can get, and also we can access to the world-class technologies that, for example, like Professor Levin or Professor Lee and Professor Cohen addressed before. And we also bring together a skilled workforce to work in collaboration, because now we can see with a clinical trial or even with a R&D program for an investigational product, it's not only scientists, clinicians to work on it, but also statisticians, bioengineers, AI scientists, data analytics expert, everyone contributing into the clinical trial program. And secondly, so as a developer, a drug developer and manufacturer, we have a rich data set of, let's say, biomedical data. So then from cross-sectoral collaboration, we can combine with the clinical data that hospitals, they are owning in order to expedite clinical trials, and also to expedite drug development to bring it to life earlier. Some of the examples that in AstraZeneca globally, we are partnering with some genomic laboratories from the universities and also from the institution. For example, like the AI Innovation Center of Karolinska Institute in Sweden, or in the UK, we have a partnership with the Francis Crick Institute, or with the Cancer Research UK in order to expedite it. Secondly would be on the partnership with the world foremost cancer centers in order to accelerate translational research, because we have the data, we have the products with mechanism, but then what we need from the clinical practice community would be how we translate it into the need of the patient. So some other partnership we established across the world, including with Johns Hopkins Medical Center, with Princess Margaret Cancer Center in Canada, and also with some other cancers, which are cancer center, which are well known in the world. And last but not least, we partner with device company in order to develop new and scalable diagnostic testing, because those support in precision medicine, especially in the phase that we bring it into life. So with biomarkers, with testing, we can identify the most appropriate patient that need the treatment.
Well, that's fantastic. I think if it's okay with you, Professor Levine, we're going to move on to the next question. And so Professor Vu and Professor Cohen, how do you envision the future in terms of smart factories? Do you envision that those will be aided by artificial intelligence and that perhaps that can augment the existing capacity of the global bioindustry in developing anti-cancer therapies? So that was a long question, but really want to know is what's the role of AI in the global bioindustry? And maybe, would you like to start us off?
Sure. So I think that, as I said earlier, that the power of AI and machine learning is predicated on the quality of the algorithm and the amount of data that's being analyzed to really train it. And so if we think about how to automate this process on a larger scale, we need to also create national data registries of tumor and blood where we can have a better idea of, from a national standpoint, where are locations where there's high prevalence of specific tumors, and how we can think about using omics and other platforms then for better biomarker analysis, diagnostics, and early detection. And I think those capabilities will give us much better opportunity to really have a more global impact.
Yeah, so that idea of the digital medical records, I don't know if you want to expand on that or give us another perspective.
Yes, so what I second what Professor Cohen said is that the foundation step is that we need to have a very respectable database, the unbiased and clean one that we can rely on, and that is first step we have to do. And I think actually this is a lacking in most of the countries, even developed countries, and in Vietnam it is a very serious problem that in the hospital, not very few hospitals have been totally digitalized, and the data system are not linked to each other yet. So if we are talking about a very aggressive advanced application of data science and AI, then we first have to establish this very foundational data system, and that's what Professor Cohen also said.
Yeah, I completely agree, and I think it's important to note that there's many developed countries that don't have a digitized system as well, so there's a lot for all of us around the world to do. Okay, well, it's now time to change to topic number 3. So this is the last set of questions. It's the shortest one, so I hope you in the audience will have your questions ready for us. In this one, we want to get the perspective of patients. How can we facilitate access to novel cancer therapies in developing countries? So, Dr. Tree, we'll start with you again. What opportunity strengths do the developing countries have in terms of developing precision cancer therapies? And how can patients from developing countries participate in clinical trials of novel cancer therapies?
Yeah, so for the opportunities and strengths, so I would see it in the angle of Vietnam in particular. So the industry, the pharma industry in Vietnam, we invest on clinical trials. And what we can see here, Vietnam has a substantial number of patients with a wide range of cancer types. There's still some similarity, but also differences between Vietnam and other countries in the world. But then working in this area, we can see most health problems, for example, like cancer burden and the determinants of the cancer burden, or local characteristic of the population in Vietnam, or even the genomics data of Vietnamese people. and responses to therapies. We don't have such kind of data available yet. So there's still a lot of problems that we can explore. The second thing that we are seeing would be in Vietnam, we have a healthcare system that is working very smoothly. And we do have some large and specialized hospitals in Vietnam, mainly for cancer. For example, like the National Cancer Hospitals in Hanoi and also the Ho Chi Minh Oncology Hospitals. And even for hematology, like hematological cancer, we do have specialized hospital for it. So it supports a lot on running the clinical trials because we have centralized patient and we have a centralized database to build on. The third one would be we have a set of regulation in Vietnam for clinical trials and has been implemented for years. And with that set of regulation, we are confident that we are reaching to the universal ethical standards in clinical trials. There's still some gaps that we can think of to motivate patients to participate in clinical trial. One of it would be increasing the awareness of the community and the patient on clinical trials. And it's not only on the patient, it's also on healthcare professional as well. Because if people see clinical trial as a means to expand their accessibility to innovative medicine, so that is a way that we can think of how clinical trial would bring support to the patient. And secondly would be on, I would say, increasing patient eligibility in order to participate into the clinical trials. We are not similar to developed countries that most of innovative medicine are available in Vietnam. There's still some not available at this moment. Secondly would be on the standard of care. So for some clinical trials, we put in a very high standard of care coming from developed country, and that would be some obstacles for patients in Vietnam, where we are not able to access to those kind of new medicine or new modalities. And lastly would be on diagnostic facilities. We should think of how we improve the diagnostics infrastructure. So with precision medicine, biomarkers and testing would be very key to identify eligible patient, and that would be something we can think of for improving the participation of Vietnamese patients in clinical trials.
Well, that was a very comprehensive answer. Thank you. And I think just picking up on your last point of diagnosis, I think we all recognize that diagnosing a cancer early really usually is indicative of how successful the treatment's going to be. And so that's a really critical point. Okay, so last question before we open it up to the audience is for Professor Levine and Professor Lee. What can we do to facilitate the access of patients in developing countries to novel but highly expensive immunotherapies?
Well, maybe I'll start with what I was talking about, the work that we're doing with the company in India and the alliance between Penn Medicine and Vin University. We have to work together to disseminate technology, and we learn from the enhancement and the improvement of access from developing countries. The other aspect, briefly, I'd like to touch on it is the education aspect and the power of the patient voice. Now, you saw Emily Whitehead in the opening movie, and I showed her picture. Her parents have started a philanthropic foundation to serve as a resource to inquiries from patients around the world about this therapy, about access to clinical trials. So in thinking about how you increase knowledge and increase access to clinical trials, I would ask, is it possible for a patient or a patient advocate to communicate with other patients? Because It's who do you trust? And for someone that has been through that experience, diagnosis and treatment, to serve as a resource, to serve as that communication channel with patients, I think provides some significant advantages.
Yeah, I think that's just a fantastic point too. I think it's very confusing for patients to figure out where to go. If there's more than one clinical trial, where do they go? What do they do? How do they access it? How do they even understand what's going on? So Professor Lee.
Yeah, in reality, this question is very important in this symposium because I found that a lot of VIP persons in Vietnam participate in this symposium. In clinical practice, in developing country as well as developed country except the United States, is very important to this kind of one. Of course, it is possible to use this new innovative treatment by clinical trial, but the number is very limited. Yeah. So in order to use broadly this expensive, innovative treatment, the first government needed the administrative improvement so that patient can easily access this treatment, and supported by medical reimbursement will be needed. In addition, active treatment and education of medical staff will be needed for future broadly use this kind of treatment in Vietnam. Also in Korea, the same situation.
Okay, well, thank you very much. So I've got to ask all of my questions, and now we welcome questions from you, the audience. So you've got the world-leading experts in this field on this panel. So please don't be shy. Yes. Can we have a microphone?
Yeah, the microphone is coming to you. Just give us 30 seconds or less.
And maybe just introduce yourself, please.
Good afternoon. My name is Sui, and I'm from the Department of Medical Genetics, VinMec Healthcare System. So first of all, thank you all the panelists for a very insightful discussion. I have my question regarding to omics technologies. You mentioned the emerging role of omics technology, genomic transcriptomics in cancer therapy. Despite of the technology advancement, when implementing it, there are obviously many different aspects to consider. So in your opinion, what would you think the most suitable strategy to implement omics technology in order to promote and to increase the efficacies of cancer treatment, and especially in immune cell therapy for the limiting settings like Vietnam and other developing countries? Thank you.
You want to start?
Well, maybe I'll start, and I can answer specifically to what we're speaking about in immunotherapies this afternoon, which is when you go to a hospital or your doctor and you get a blood test, what do they measure? They measure chemistries, they measure some very simple complete blood count, maybe some subsets. But what we see with the development and administration of these therapies is a need for an immune health assessment by various omics technologies. When a patient comes in, how do we know how they will respond, if they will respond, if we don't have this information, this detailed information on their immune health? And in that way, we can think of precision biomarkers and perhaps a partial way to address the cost question, because we may be able to predict patients who are more likely to respond to these immunotherapies, or patients who are not likely to respond, then they would be directed towards other therapies. So I'll turn over perhaps to other panelists to add their perspective.
So I would just add that I think that when you look at application of omics in both diagnostic and therapeutic setting, you have to also look at what are the capabilities for some new biomarker analysis from RNA platforms as well as microRNAs, EVs, and other types of blood markers. So this way we can use these combination omics platforms and develop better AI from that data set to really understand tumor resistance, understand combination therapy strategies, and be more precise for individual patient both diagnosis and treatment. So,
Thank you. Anybody else? Okay, well, I hope that answered your question.
Very much.
Thank you.
We have several hands up,
Okay.
at least 4.
Hi, I think I've got a mic. Hi, I'm John Nost. I was one of the earlier speakers on innovation and digital transformation. Question is there any path
Sorry,
forward?
can you just come forward? We've got these lights blaring in our eyes, so we can't really, you're sort of a shadow.
I like to hide behind the light.
Okay. Thank you so much.
Is there a path forward on off-the-shelf or allogenic versus autologous therapy, particularly with an eye on cost?
Yeah, it's a great question.
So I'll start. There are a number of academic centers and companies developing allogeneic cell therapies of various types. One approach using CAR T cells or CAR NK cells is to knock out genes that may cause rejection, specifically T cell receptor and the human leukocyte antigen complex. But what remains are minor antigens that are still seen by the immune system. So one can prolong the engraftment and persistence, but not to the level of autologous cell therapies. Early on, the pitch of the allogeneic cell therapies was we can make many, many hundreds, maybe thousands, who knows how many doses from one single patient, but that doesn't really seem to be true. Because to get that number, you have to grow the cells longer. When you grow the cells longer, they get more exhausted, and when they get more exhausted, they get less potent. Another strategy with potential is induced pluripotent stem cells differentiated to NK cells or macrophages or T cells, and those also are in clinical trials. And I would say there are some early indicators of clinical responses, but the jury is still out. And then a third is what I mentioned in the presentation in one brief slide, which is the potential ability to generate these redirected T cell or NK cell therapies in the body by delivering nanoparticles, perhaps lipid nanoparticles or other types of vectors to re-educate the immune system in the body.
Yeah, thank you for that very comprehensive answer. Did you want to add anything to that?
Yeah, allogenic versus autologous, there are the advantage and disadvantage for each the cell type. So in case of autologous cell, I'm a hematologist, so in case of hematologist, it's very easily think about autologous treatment because we already have a big experience for stem cell transplantation. In case of allogenic, we can make the off-the-shelf. That is most the merit point. But there is possibility of some kind of rejection after repeated treatment. And there is some possibility of some side effects as like the GVHD. But in NK cell, it's not the different story. In case of autologous stem cell, the autologous stem cell have the personalized, we can make personalized cell, but there is some slightly less effect than initially because the allogenic cell have a allo-reactivity to treatment. In the long time of story, I favorite the autologous stem cell because I'm a hematologist. But in company, in the aspect company, absolutely favorite to allogenic stem cell to make off-the-shelf as like drug.
Let me add one more point, which is there clearly is a need because there are patients from whom we cannot obtain or generate autologous cell therapies. Some patients who have been very heavily treated or infant ALL. So I hope that this continues, and people often ask, when will off-the-shelf replace autologous? And I think the answer is not anytime soon, but there probably will be some coexistence.
Yeah, and that's a fantastic question because I think that's the hope and the dream that we're all striving for, because something that's off the shelf would be so much less expensive to manufacture. But clearly, cell therapy is the most expensive, medical treatment to manufacture, just because you're dealing with living cells. Okay, thank you for those excellent questions.
We have a number
yes,
of
we have
questions.
lots of time for more questions. Thank you. There's a question over here. Oh. Thanks. Could you just introduce yourself?
I just talk. Yeah. I'm Jayanti Anandan. My question is, when does this therapy work? At what stage of cancer? Supposing the cancer is not detected till the third stage, for the silent cancer like ovarian cancer, these are not even coming to the knowledge of the patients. So when is the stage where this therapy will be more effective? Thank you.
Yeah, so maybe I'll start. And as with most new therapies, these are first tested in patients who have exhausted all available options. And that was the case in acute lymphoid leukemia, in lymphoma, and in multiple myeloma. Now there have been trials in second-line lymphoma and also earlier lines of other blood cancers. And a year ago, 2 of those clinical trials demonstrated superiority to standard of care in second-line lymphoma. So now they're being integrated. Yes, in earlier lines, these therapies would probably be more effective, but then we have the flip side of it, which is, can we deliver these therapies? Can we manufacture them at scale? Particularly for autologous, it's a huge challenge. And that's why it's so important to think about how we streamline this, both autologous, maybe iPSC, and eventually in vivo.
Yeah, and in your question, you highlighted ovarian cancer, and I think that comes back to the question of diagnosis, and can we detect these things? We were talking earlier about these being kind of silent cancers. Did you want to add something?
Oh, no, yeah, I just have a note here, like we talk a lot about treatment, and when treatment comes, it is either painful or expensive, and usually both. So, and we talk a lot about application of AI and machine learning there, and one of the applications we can think of is to have early detection of cancer, and that's very important. That reduces the cost and the pain of the patient by a factor of 10 easily. And that's where AI and statistical tools can be very useful because they are very good at detecting, pattern. And when you see the pattern, you already have a suspect, like, okay, this guy have a good chance to develop cancer. And then you follow those guys. And that is the direction I like to promote in Vietnam because we have a lot of patients and we detect them at very late stage. And that make it very, very difficult for treatment. So Levit Group has a spin-off called Gene Story, actually, is the company doing exactly that. So they go into the direction of early detection rather than treatment. And that is something we may want to consider.
Yeah, that's fantastic. Did you want to add?
Yeah, I just wanted to add, like for many solid tumors with early detection, if you find a tumor at early stage, it can be curable oftentimes with surgery. Whereas once it becomes systemic or metastatic, that's when issue of what therapy is the best. So understanding earlier when that metastatic spread is happening to really align patients with therapy options would be an area where AI and machine learning and omics can really help.
Did you want to add?
That kind of question is very important in the field of hematologic malignancy. So because hematologic malignancy, especially multiple myeloma, is so difficult to cure. So for more time, we focus to try to treat the late stage of cancer, relapsed refractory patient. But it's also, we investigated near line. It's so difficult to cure in the final line. So you are focusing to early stage of cancer, we call smoldering multiple myeloma. That means not developed the symptom, you have the bone marrow myeloma cell. So investigators try to put strong treatment to the early stage of cancer for cure, increase the cure fraction. So at this time, most of the clinical trial perform the late stage of cancer. I already mentioned the Professor Levine. But in the future, we will focus to this clinical trial from late stage to early stage to increase the cure fraction.
Thank you very much.
Thank you. Right here in the front. Could we have the microphone in the front? Oh, over here. Thank you.
I am Dr. Lim from VinFuture Research, the Stem Cell Institute. I have one question for Professor Bruce Levi and one question for Professor Jung Lee. The first question is, so far we know the success rate of CAR T cell is around 60 or 70%. So in the 30 or 40%, how can cancer cell escape from CAR T cell? Otherwise, what is the mechanism? And second question for Professor Jung Lee is, do you think the allogeneic NK is better than autologous NK? Because in a cancer patient, autologous NK cell already fail to prevent cancer development. Thank you.
Thank you, Professor, for the question on success rate. In pediatric acute lymphoid leukemia, there is a very high success rate, even in relapse refractory patients, that is around 90%. In lymphoma and myeloma, it's lower at around 50%. So why is that? Primarily, there are tumor intrinsic factors. Perhaps there's an escape mutant, or perhaps there is severe immunosuppression in the tumor microenvironment. There's also the other side, which is the immune factor. Do we have a patient who has undergone so many previous therapies where his T lymphocytes are of poor quality? Well, how can we improve that quality? That's why I mentioned, how do we teach every T cell all that it can be? What factors, what changes in the manufacturing, what genes can we add to enhance the potency of the T cells or the NK cells for that matter? And that's where what we were talking about in the earlier question of diagnosing immune health, and the integration of AI will help. Because we have, by my count over the past 2+ years, identified maybe 25 to 30 methods to improve T cell potency. Well, that's a large number. If we think of a combination, 2 to the 25th, it's a very large number, right? But that also points to the importance of having many groups around the world work on the development of these therapies and enhancing potency so that we increase the success rate, because it's only by doing parallel processing that we gain the most information in the fastest manner.
Yeah, for the actually, that is very good question and comment. I completely agree. The limitation of autologous NK cell. So most limitation of autologous NK cell is the dysfunctional NK cell in cancer patient you already mentioned. So that kind of one is the effect to generation of autologous NK cell. Some patients have a problem to generate NK cell to treat the cancer. But if we use just NK cell isolated from patient, that's a problem. But we can make NK cell activation and expansion. That kind of activated and expanded NK cell have strong effect compared to the naive NK cell. So that we can overcome this kind of disadvantage by autologous NK cell.
Well, we're almost time, but if somebody has a quick question. Okay, one last quick question.
Can the mic go to the gentleman?
Maybe, okay, go ahead.
I'm Dr. Chu, I'm hematologist from VinMed Hospital. My question is that, one of the limitations of CAR-T therapy is that it takes time to make the final product. Usually it takes 4 months to have the final CAR-T product to use. But the patient at the relapse or refractory stage usually don't have enough time, especially in the developing country like Vietnam. It takes up to half year to can have the product. So we are focusing on another kind to approach for these patients by specific antibody. So they also have the same mechanism with the CAR-T therapy, and they are available and more realistic to invest in current condition in Vietnam. So how do you think about the future of bispecific antibody compared with the CAR-T therapy for using patients in developing country like Vietnam? Thank you.
Well, thank you.
Can we just have a quick answer? I know
Yes.
it's a very great question.
Yes, I can answer very quickly that we have some success with bispecific, even trispecific antibodies, but still we have patients that relapse and don't respond that we're able to treat with CAR T cells, albeit more expensively. So how do we learn from each other and, again, improve the efficiency of these immunotherapies?
Well, thank you. And you answer, is that okay? Or did you want to add something very quickly?
Can I edit for the question? It's the same story in Korea. Yeah. We have CAR T-cell for clinical trial, bispecific antibody and antibody drug conjugate. So everybody consider in the past CAR T, bispecific antibody and antibody drug conjugate is much better than CAR T-cell, but everybody think CAR T-cell is with single treatment, and there is no need for repeated injection like that, compared to bispecific and ADC. If we handle in Korean society investigator, previously ADC and bispecific are favorite. Now CAR T-cell is favorite. But in Vietnam, I think the bispecific and ADC is much more important before entering CAR T-cell therapy.
Well, thank you all very much for those amazing questions. And if you didn't have a chance to ask questions, we'll be around. So please ask questions. So I just wanted to make a couple of concluding remarks. We see enormous potential and possibilities at the intersection of different disciplines, as highlighted by our panelists here today, with expertise in AI and big data, immunology, models of disease, and from different sectors of industry and academia. Clearly, to make a difference, we have to learn each other's language in order to communicate effectively. And we have to take advantage of all of our collective creativity to bring about change. We know that change doesn't just happen. We rely on the creativity of our private and public sectors, the scientists, clinicians, government bureaucrats, and administrators, to have the courage to do things differently. And of course, we are grateful to our patients who enter into clinical trials not knowing the outcome and not even knowing whether they've been given the treatment or the placebo. So with this, we have the last remarks given to Professor Levine and Professor Cohen. What areas of preclinical and clinical research need to be further explored? And what government policies are required to elevate the success of cancer research and treatment?
So I think, as was said a little bit earlier by several of the panelists, as we think about some policies and opportunities, if we could create a better national resource or biorepository where we have databases for cancer patients, this can give an incredible opportunity to build AI and machine learning opportunities to really predict disease diagnosis earlier, to think about therapy response, and also to understand resistance risk. And investing in this type of infrastructure can certainly advance the ability of us to do the combined collaborative research that's going to lead CAR-T and other therapies for patients.
And I would add, across all of these therapies, whether they're CAR-T or NK or bispecifics, the conduct of clinical trials, the enrollment in patients in clinical trials, the access of patients to clinical trials speeds the development for new medicines, because the faster we get an answer as to whether something looks promising, the faster we can move on to develop it or to move in another direction. As far as government policies, I think it would be to look at the existing regulations that in many cases may be written, or most cases written in the era of small molecules and even biologics. And these therapies are very different. I made a list of some of the testing that we do, but it's very different to think about potency for a small molecule than it is to think about potency for a living drug. So how do we adapt the framework of regulations to adapt to these new therapies?
Thank you very, very much, our panel of scientists, and thank you, this session's chair, Professor Molly Choquette. We had a wonderful discussion. Thank you for all the great questions coming from our audience today. And through your valuable expertise, we have learned updates on precision cancer therapy worldwide and the opportunities that exist for Vietnam. On behalf of the organizers, we would like to express appreciation for your attendance as well as your support to VinFuture Prize 2022, which honors science and technology that serve humanity. Thank you very much and have a great afternoon and evening. Thank you.